Gregor Johann Mendel played a vital role in discovering the laws of heredity. He carried out several hybridization experiments with true breeding pea lines which had sharply contrasting characters. One such experiment was the crossing of a tall and a dwarf pea plant. Since the cross was between plants that differed in a single trait, that is, height. In this case, Mendel called it a monohybrid cross. Furthermore, Mendel called the tall and the dwarf plants in this experiment the parental or the P generation. After the cross between the tall and dwarf plants, Mendel collected the seeds and planted them to produce the first hybrid called the F1 generation, or the first filial of plants. He found that all the plants in the F1 generation were always tall, like one of the parents, and the dwarfness had disappeared. Mendel conducted similar experiments with other pairs of traits and made certain observations. Every single time, he found that the F1 hybrid resembled only one of the parents, while the other parent's trait was missing. To explore this further, Mendel self-pollinated the F1 hybrids to produce a second hybrid generation of plants, known as the F2 generation, or second filial of plants. Interestingly, this time, the progeny included both tall and dwarf plants in a definite ratio. 3/4 of the plants in the F2 hybrid were tall, while 1/4 was dwarf. Mendel also noticed that the plants did not show blending or mixing. That is, none of them were of in-between height. Once again, to confirm his observations, Mendel conducted experiments with other traits. His results were confirmed when he found that both the parental traits were expressed in the F2 hybrid in the proportion 3:1. Moreover, the contrasting traits did not show any blending in either the F1 or F2 stages. From his experiments, Mendel inferred that something was being stably passed on from parents to progeny over successive generations. This was the reason the dwarfness that had disappeared in the F1 hybrid had reappeared in the F2 hybrid. Mendel called this something factors, which are known as genes today. Genes are represented by letters for our convenience. The trait expressed in the F1 hybrid is denoted by a capital letter, while the other trait is denoted by a small letter. As all plants obtained in the cross between tall and dwarf plants were tall in the F1 hybrid, the tall trait is denoted by capital T and the dwarf by small t. Here, capital T and small t are alleles of each other. That is, they are a pair of genes representing two alternative forms of the same character, namely the height of the plant. Therefore, pairs of alleles for height in plants would be capital T, small t, and small t, small t. Here, capital T, capital T, small t, and small t are genotypes of the plant, and the descriptive terms tall and dwarf are the phenotypes. The alleles in the genotype capital T and small t are similar or homozygous, while the alleles in the genotype T small t are dissimilar or heterozygous. Moreover, Mendel also called the character that expresses itself in the F1 hybrid dominant, and the one that failed to express itself recessive. Therefore, through his experiments with tall and short pea plants, Mendel found that the character of tallness was more potent over the character of dwarfness in the F1 hybrid, and that both tallness and dwarfness appeared in a 3:1 ratio in the F2 hybrid. Mendel called this the law of dominance. The law states that in a monohybrid pure line cross between parents with contrasting traits, only one form of the trait, that is, the dominant form, appears in the F1 generation, and both forms appear in a ratio of 3:1 in the F2 generation. Also, from his experiments, Mendel observed that the recessive parental trait was expressed without any blending in the F2 hybrid. After verifying the results of the crossings, it was found that the segregation of alleles is a random process, and the chances of a gamete containing either allele is 50%. This is known as the law of segregation, and it states that alleles do not show any blending, but instead segregate from each other during gamete formation into different gametes. Both the characters governed by these alleles are recovered in the F2 generation, though one of the characters is not displayed in the F1 generation. A diagram that is very useful in predicting the outcome of a particular cross or breeding experiment is Punnett square, which was devised by British geneticist Reginald C. Punnett. Punnett square is a graphical representation that helps calculate the probability of all possible genotypes of the progeny in a genetic cross. Let's take a look at how Mendel's experiments are denoted using the Punnett square. In the F1 generation, Mendel crossed homozygous dominant T parent with a homozygous recessive small t parent. The gametes produced by the parents are written on two sides of the square, and the T progeny produced by the fertilization of the gametes is written within the square. Though the genotype of the F1 hybrid is T, its phenotypic character is tall. Plants of the genotype T produced by the F1 hybrid are then self-pollinated to produce gametes of the genotype T and t in equal proportion. From the square, you can easily see that the pollen grains of genotype T have a 50% chance of pollinating the eggs of the genotype T as well as t during fertilization. Similarly, pollen grains of genotype t have a 50% chance of pollinating eggs of genotype T as well as t during fertilization. Thus, random fertilization results in zygotes that are of genotypes T, T and t. From the Punnett square, you can also see that 1/4 of the random fertilizations have led to T, half has led to T, and finally 1/4 has led to t. Therefore, the genotypic ratio is 1:2:1. And as 3/4 of the plants are tall and 1/4 dwarf, the phenotypic ratio is 3:1. The ratio 1:2:1 or 1/4:1/2:1/4 of capital T is to capital T small t is to small t is condensable to a binomial expression of the form ax plus by the whole square. The expression has the gametes bearing the genes capital T or small t in equal frequency of 1/2. This expression can be expanded as shown. Thus, we can calculate genotypic ratios using mathematical probability. However, by just looking at the phenotype of a plant, it is not possible to predict its genotype. For example, you cannot predict whether a tall plant from the F1 or F2 hybrid is homozygous dominant, that is, capital T, or is heterozygous dominant, that is, capital T, small t. Therefore, a test cross or a cross between individuals of the F1 generation with a homozygous recessive parent is carried out to determine whether the genotype of the individual in question is homozygous dominant or heterozygous dominant. Thus, through his monohybrid cross experiments, Mendel proposed the laws of dominance and segregation. This is little Sam. Can you see his face is swollen? Moreover, his eyes are slanting and bulging, and he has a small mouth with a protruding tongue. These are a few symptoms of a chromosomal disorder known as Down's syndrome. Normal human beings have 46 chromosomes arranged in 23 pairs. However, even a slight variation from this pattern causes abnormalities or disorders. Chromosomal disorders are caused either due to changes in chromosomal number or changes in chromosomal structure. Changes in chromosomal number occur due to non-disjunction of chromosomes, which is the failure of chromatids to disjoin during cell division, leading to either aneuploidy or euploidy. Aneuploidy is a condition where one or more chromosomes are either gained or lost. Aneuploidy is of two types, trisomy and monosomy. A normal diploid individual has 2n number of chromosomes. However, when an extra chromosome is added, the condition is known as trisomy, that is, a 2n plus 1 condition. On the other hand, when a chromosome is lost or when a chromosome of any pair is absent, the condition is known as monosomy, that is, a 2n minus 1 condition. Now, euploidy is a polyploidic condition, where more than two haploid sets of chromosomes are formed due to the failure of cytokinesis after the telophase stage. Depending on the number of chromosomal sets added, euploidy can be triploidy or a 3n condition, tetraploidy or a 4n condition, pentaploidy or a 5n condition, and so on. Let's now take a look at some chromosomal disorders that occur due to changes in chromosomal number. Down's syndrome or trisomy 21 is caused due to the presence of an extra copy of the 21st chromosome. It was Dr. Langdon Down, a British doctor, who first described this disorder, and hence the name Down's syndrome. The symptoms are a swollen face, bulging and slanting eyes, a small mouth, and a protruding and furrowed tongue. Some other common symptoms of this disorder are short stature, small round head, a broad palm with a single crease, and retarded physical, psychomotor, and mental development. Another disorder caused by changes in chromosomal number is Klinefelter's syndrome. It is a condition where males have an additional X chromosome, which results in a karyotype of 47, that is, XXY. Klinefelter's syndrome is also known as XXY syndrome and is named after Dr. Harry Klinefelter, who was the first to describe the disorder. It is one of the most common sex chromosome disorders in males and shows symptoms such as poor beard growth, narrow shoulders, gynecomastia or breast development, and underdeveloped testis. Moreover, such males are sterile. Yet another disorder due to changes in chromosomal number is Turner's syndrome, where one of the X chromosomes is absent in females. Chromosomes in these females will be 45 with XO. The syndrome is named after Henry Hubert Turner, an endocrinologist who first described it. Females with this syndrome show symptoms like a webbed neck, constriction of the aorta, poor breast development, underdeveloped ovaries, and short stature. Moreover, such females are usually sterile and have health problems like diabetes, hearing and vision problems, and hypothyroidism. Just like changes in chromosomal number, any change in the structure or arrangement of genes can also lead to chromosomal disorders. Changes in chromosomal structure can take several forms. Some of these changes include a portion of a chromosome getting deleted, duplicated, or transferred to another chromosome. While some of these structural changes in a chromosome are inherited, others take place accidentally when reproductive cells are being formed or during early fetal development. Jacobson syndrome and Kryder-Katt syndrome are some disorders caused due to changes in chromosomal structure. Therefore, chromosomes hold the genetic keys to all the functions of our body, and any change in the number or structure of a chromosome can lead to chromosomal disorders. Aren't these flowers beautiful? These are snapdragon flowers, and they come in so many different colors. Hybridization experiments conducted on pea plants were conducted on snapdragon plants too. Apart from snapdragon plants, experiments were conducted on birds like pigeons and poultry, and on several animals like mice, rabbits, cats, guinea pigs, and even man. However, it was discovered that the law of dominance didn't hold true in all cases. That is, Mendel's laws were not universal in occurrence. To understand this better, let's further study the experiment conducted on the snapdragon plants. When a true breeding or a pure line red-flowered snapdragon with genotype R was crossed with a true breeding white-flowered snapdragon with genotype r, the results were quite different from what was seen in the case of the pea plants. The flowers of the F1 hybrid were pink, and the genotype was R. That is, the F1 hybrid flowers had a phenotype that resembled neither of the parents, but was in between the two. Now, the flowers of the F1 hybrid were self-pollinated, and the genotypic ratios obtained in the F2 hybrid were 1:2:1, the same result obtained by Mendel in his experiments on peas. However, the phenotypic ratios obtained in the snapdragon plant, as you can see, is also 1:2:1, which is different from the ratio 3:1 dominant is to recessive ratio in the pea plant. So, in the case of incomplete dominance, both genotypic and phenotypic ratios are the same. That is, 1:2:1. Thus, when a dominant allele does not completely mask the phenotypic expression of the recessive allele in a heterozygote, a blending of both dominant and recessive traits takes place in the F1 and F2 heterozygotes. This phenomenon is called incomplete dominance. This is why the phenotypic ratios obtained in the experiment on snapdragon plants different from those in the pea plants. It is the gene for flower color that controls the amount of pigment in petals. Each allele is a code for a trait, that is, specific amount of pigment. When both alleles for pigment are present, the petals are dark red due to heavy pigment production. On the other hand, if none of the alleles for pigment exist, the flower is white. However, when only one of the alleles is present, only half the pigment is produced, creating a pink shade. The alleles are therefore exhibiting incomplete dominance, which is a form of intermediate inheritance where one allele for a specific trait is not completely dominant over the other allele. But why is it that some alleles are dominant while others are recessive? Let's understand this by studying the functioning of a gene in greater detail. As you know, a gene is the unit of heredity that occupies a fixed position on a chromosome called the locus. A gene generally has two forms called alleles. In homozygotes, the alleles are the same, while in heterozygotes, they are not identical. An allele may be different due to certain changes it has undergone, which modify the information contained by that particular allele. For example, Let's consider a gene that contains information for producing an enzyme. This gene contains two copies, which are the two allelic forms. Now, let's assume that the normal allele produces the normal enzyme that is required to transform a substrate S. According to assumed facts, the modified allele could be responsible for producing a normal enzyme, a less efficient enzyme, a non-functional enzyme, or no enzyme at all. Now, if the modified allele produces a normal or a less efficient enzyme, we can say that the modified allele is equivalent to the unmodified or normal allele. That is, the modified allele will produce the same phenotype or trait, or in other words, it will bring about a transformation in the substrate S. Such equivalent allele pairs are quite common. However, if the modified allele produces a non-functional enzyme or no enzyme, then the phenotype is dependent on the functioning of the unmodified or the normal allele. Here, the unmodified functioning allele or the normal allele that represents the original phenotype is the dominant allele, and the modified allele is generally the recessive allele. So, in the example we just saw, the recessive trait was due to a non-functional enzyme or because no enzyme was produced. Therefore, incomplete dominance causes a distortion of the normal phenotypic ratio and results in offspring with a combined phenotype. This is a homozygous bull, white in color, with a genotype W, and this is a homozygous cow, red in color, with a genotype R. What color will their calf be in the F1 generation? Well, it isn't white or red, and neither is it a color in between. The calf is roan-colored. Its coat is a mix of red and white hair. Therefore, in this case, the F1 generation doesn't resemble either of the parents as in dominance, nor is it in between as in incomplete dominance. What the calf displayed was codominance. Codominance refers to a situation where a heterozygous organism has a phenotype that demonstrates traits from both dominant as well as recessive genes. A codominant trait is not blended but is independently and equally expressed. Another widely known example of codominance in human beings is the ABO blood system, which is used to determine human blood type. The plasma membrane of the red blood cells, or the RBCs, present in human blood, has sugar polymers that protrude from its surface. It is these sugar polymers that determine the specificity of the major blood types. For example, the difference between blood types A and B lie in a single sugar unit that protrudes from the end of a carbohydrate chain of a glycoprotein or glycolipid on the plasma membrane of an RBC. It is the gene I that is responsible for determining the type of sugar produced by the RBCs. This gene has three different alleles, namely IA, IB, and small I. Of these, alleles IA and IB produce sugars that are slightly different from each other. while the allele small I doesn't produce any sugar. Here, the alleles capital IA and capital IB are completely dominant over allele small I. Now, human beings are diploid organisms and hence possess only 2 of the 3 alleles or the gene I. Consequently, as you can see, there are 6 possible combinations of alleles. Therefore, if the allele from one of the parents is capital IA and from the other is small I, only capital IA is expressed in the offspring, as small I does not produce any sugar. In this case, the RBCs have only type A sugar in them, and the genotype of the offspring is capital IA small I, and the phenotype or the blood group is A. Similarly, when the allele from one of the parents is capital IB and from the other is small I, only capital IB is expressed in the offspring, as the RBCs have only type B sugar in them. In this case, the genotype is capital IB small I, and the phenotype is B. However, when the allele from one parent is capital IA and from the other is capital IB, both the alleles express their own types of sugar in the offspring due to codominance. In this case, the RBCs have both A and B types of sugars in them, and the genotype of the offspring is capital IA, capital IB, and the phenotype or the blood group is AB. Now, if the alleles from both parents are capital IA, once again the RBCs have only type A sugar in them, and therefore the offspring has a genotype capital IA, capital IA, and phenotype A. Likewise, if the alleles from both parents are capital IB, the RBCs have only type B sugar, and so the offspring has a genotype capital IB, and phenotype B. However, if the alleles from both parents are small I, the RBCs have no sugar in them, and so the genotype of the offspring is small I, and the phenotype is O. Therefore, as there are 3 different types of alleles, there are 6 possible genotypes and 4 different blood groups or phenotypes that exist. Apart from being a good example for codominance, the ABO blood grouping system is also an ideal example of multiple alleles. As we just saw, the gene I has 3 alleles, capital IA, capital IB, and small I, that govern the same character, that is, blood group. However, since an individual can have only 2 alleles, it is possible to learn about multiple alleles only when detailed population studies are conducted. According to the multiple alleles concept, more than 2 alleles govern the same character. Now, let us study an experiment that shows how a single gene may sometimes produce more than one effect. Pea seeds have a single gene with 2 alleles, capital B and small b, that control starch synthesis in them. In the experiment conducted, effective starch synthesis was seen in capital B homozygotes, and the seeds produced were large or had a large amount of starch in them. Moreover, the pea seeds were round. On the other hand, small b homozygotes were not very efficient at synthesizing starch, and so the pea seeds produced were small or had less starch in them. Also, the seeds were wrinkled. Moreover, in the experiment conducted, the seeds or the starch grains produced by heterozygotes B were of intermediate size but were round. Now, in this experiment, if we consider the starch size as the phenotype, then the alleles are displaying incomplete dominance. On the other hand, if we consider the shape of the seed as the phenotype, then the alleles are displaying dominance, as the seeds were round like one of the parents. Hence, the experiment shows that dominance is not an autonomous feature of a gene or gene product. Therefore, dominance, incomplete dominance, or codominance depends on gene product and the production of a particular phenotype from this product. It also depends on the phenotype that we wish to examine in case the same gene influences more than one phenotype. Did you know that Gregor Mendel cultivated and tested about 29,000 pea plants between 1856 and 1863? One such test was the crossing of pea plants with yellow and round seeds and those with green and wrinkled seeds. As you know, such a cross between plants that differ in two traits or characters is known as a dihybrid cross. This experiment, conducted by Mendel, resulted in a pea plant that produced yellow and round seeds in the F1 generation. Here, can you guess the dominant shape and color? Obviously, the round shape is dominant over the wrinkled shape and the yellow color dominant over green. Mendel noticed that these results were identical to the results he had obtained when he had carried out monohybrid crosses between plants with round and wrinkled seeds and those with yellow and green seeds. Now, in Mendel's dihybrid experiment, let's denote the dominant yellow seed color with the genotypic symbol capital Y and the recessive green seed color with small y. Likewise, let's denote the round seed as capital R and the wrinkled seed as small r. The genotype of the parents can therefore be denoted as capital R, capital Y, and small r, small y, small y. On crossing the plants, the gametes capital R, capital Y and small r, small y unite to produce yellow and round seeds with the genotype capital R, capital Y, small y in the F1 generation. Mendel further self-pollinated the plants obtained in the F1 generation to produce F2 generation plants. This time, he found that 3/4 of the seeds were yellow, while 1/4 was green. Similarly, he noticed that 3/4 of the seeds were round, while 1/4 was wrinkled. Therefore, the yellow and green color had segregated in the ratio 3:1, similar to the results obtained in a monohybrid cross. And the round and wrinkled shape had segregated in the same ratio 3:1. Mendel also found that the phenotypic ratio in the F2 hybrid was 9:3:3:1. That is, there were 9 round yellow seeds, 3 round green seeds, 3 wrinkled yellow seeds, and 1 wrinkled green seed. Mendel had got the same phenotypic ratio in several dihybrid experiments that he had conducted. This ratio 9:3:3:1 can be derived as a combination series of 3 yellow is to 1 green with 3 round is to 1 wrinkled. And further, this derivation can be written as shown. From such observations on dihybrid crosses, Mendel proposed a new set of generalizations which was later called the law of independent assortment. According to this law, when two pairs of traits or characters are united in a hybridization experiment, the segregation of one pair of characters is independent of the other pair of characters. We can use the Punnett square to easily understand the independent segregation of the two genes during meiosis and the production of pollen grains and eggs in the F1 generation. For example, in the dihybrid cross experiment conducted by Mendel, the genotype of the seed obtained in the F1 generation was capital R, small r, capital Y, small y. Now let's consider the segregation of one pair of genes, capital R and small r. Here, 50% of the gametes have the capital R gene, and the remaining 50% have the small r gene. Now, apart from each gamete having either capital R or small r, it must also have either capital Y or small y. Here, it is important to note that the segregation of 50% capital R and 50% small r is different from the segregation of 50% capital Y and 50% small y. Hence, 50% of the gametes bearing small r has capital Y and the remaining 50% has small y. Similarly, 50% of the gametes bearing capital R has capital Y and the remaining 50% has small y. Therefore, the genotypes of the gametes are of 4 types, namely capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. In other words, there are 4 types of eggs and 4 types of pollen. And as you can see, the frequency of these gametes is 25% or 1/4 of the total number of gametes produced. Now, the allelic composition of the zygotes of the F2 plants can be easily obtained using the Punnett square as shown. As you already saw, there are 4 different types of phenotypes formed, and the phenotypic ratio is 9:3:3:1. However, 9 different genotypes result from the F2 hybrid, and the genotypic ratio is 1:2:1:2:1:2:1. Thus, from the dihybrid cross experiment, Mendel found that the gene combination of the progeny differs from the parental gene combination. Gregor Mendel, the father of modern genetics, not only researched pea plants, but also carried out several experiments with honeybees. In fact, by 1865, Mendel had published his work on inheritance of characters. However, his work did not receive due recognition until the beginning of the 20th century due to several reasons. Firstly, Mendel's work couldn't be widely publicized because means of communication in those days were not well developed as they are today. Secondly, Mendel had considered genes or factors as discrete and stable units responsible for controlling the expression of traits. He had also explained the concept of alleles and how they were responsible for the continuous variations observed in nature. However, his concepts about genes were not readily accepted by his colleagues. Thirdly, Mendel, for the first time, had applied statistical analysis and mathematical logic to problems in biology, which was objected to by many biologists of his time. Finally, Mendel could neither explain what genes were made of, nor could he provide any physical proof of their existence. Mendel's work got due credence only in 1900, when his results on the inheritance of characters were independently rediscovered by three scientists: Hugo de Vries, Carl Correns, and Erich von Schermach. Also, both chromosomes and genes segregate during the process of gamete formation, and only one of each pair is passed on to a gamete. However, they differ in the way they segregate. In genes, independent pairs segregate independently of each other, while in chromosomes, one pair segregates independently of another pair. Let's now study the behavior of chromosomes during cell division. In the anaphase stage of meiosis I, a set of chromosomes aligns along the metaphase plate. Here, you can see a long orange and a short green chromosome, as well as a long yellow and a short red chromosome aligned at the same pole. Now, consider another set of chromosomes aligned along the metaphase plate during the anaphase stage of meiosis I. Here, there is a long orange and a short red chromosome and a long yellow and a short green chromosome arranged at the same pole. Now, during the anaphase stage of meiosis II, you can see the orange and green chromosomes segregating together in the cell on the left, while the orange and red chromosomes segregate together in the cell on the right. Sutton analyzed this segregation of chromosomes during meiosis and concluded that chromosomes have individuality and they occur in pairs, with members of each pair contributed by each parent. He also concluded that the paired chromosomes separate from each other during meiosis, and the distribution of the paternal and maternal chromosomes in each homologous pair is independent of each other. Both Sutton and Boveri were of the opinion that the pairing and separation of a pair of chromosomes would result in the segregation of a pair of genes or factors carried by them. Their hypothesis on chromosomal behavior came to be known as the Boveri-Sutton chromosome theory. In fact, Sutton combined this hypothesis of chromosomal segregation and Mendelian principles and called it the chromosomal theory of inheritance.